What we had shown is that the transgenic muscle-specific Nor-1 mouse exhibited many of the features of exercise, many of the beneficial features of exercise. So the current manuscript is addressing understanding the association between the nuclear receptor Nor-1 and exercise and identifying the cellular mechanisms.
In the current manuscript, we exercise mice for 40 minutes on a treadmill. In response to that exercise, at 1 hour and 4 hours after, we showed an induction of the expression of the alpha-nuclear receptor Nor-1 in skeletal muscle. Following on from this, we wanted to discover what actually causes the induction of Nor-1, what are the pathways involved. So we looked at the involvement of calcium signaling and downstream phosphorylation targets. We showed that the induction of Nor-1 is dependent on calcium signaling, and that goes through a calcineurin dependent pathway, which then activates P38 and phospho-Krebs to ultimately induce Nor-1 expression. So we wanted to examine the molecular mechanisms involved in the induction of Nor-1 following exercise. To do this, we approached this in a few different ways, and the first way that we did is that we treated skeletal muscle cells with a calcium ionophore to mimic what happens during exercise. This also induced Nor-1 expression similar to exercise. The second key experiment we did is that we treated mice prior to exercise with cyclosporin A, which is a calcineurin inhibitor. This inhibited the expression of Nor-1 following exercise, showing that the induction of Nor-1 during exercise is calcineurin dependent. Following on from this, we showed results that exercise induces the phosphorylation of Krebs. In the literature and our previous work, we've shown that Krebs is a key transcription factor involved in the induction of Nor-1 at the Nor-1 promoter. So we then decided to look at the Nor-1 transgenic model again, and we examined the phenotype of skeletal muscle in terms of vascularization. We discovered that the skeletal muscle of the Nor-1 transgenic has increased vascularization. So if you look at figure 3, you can see that there's increased vascularization around the muscle fibers. So essentially the Nor-1 transgenic mouse has an increased network of capillaries around each muscle fiber. The other striking phenotype that we discovered in the Nor-1 transgenic model was that the skeletal muscle of the transgenic has hypertrophy. So in figure 4 we demonstrated hypertrophy in two ways. The first way is that we showed that there was increased muscle mass in different muscle types that we examined. The second thing is that in the tibialis anterior we took a cross-section and measured the cross-sectional area of each muscle fibre. This showed that the cross-sectional area of the muscle fibres was increased in the Nor-1 transgenic. So we wanted to identify the molecular mechanisms involved in the phenotype of the Nor-1 transgenic mouse model. We looked at proteins associated with autophagy. We showed that there was increased expression of proteins involved in the autophagosome membrane, so LC3A and ATG5. And we also showed that a protein involved in the cargo recycling and transport was decreased. That was P62. To further explore the connections between Nor-1 and autophagy, we used an in vitro model to examine whether Nor-1 expression induces active autophagy. We transfected Nor-1 with a GFP-RFP LC3A reporter and discovered that Nor-1 expression induces the formation of active autophagolysosomes. In the current study we showed that Nor-1 expression is induced in skeletal muscle in response to exercise, and that this induction is calcineurin dependent. In the second part of the study, Nor-1 expression drives autophagy in vitro and in vivo. Intracellular recycling is required for dramatic changes in phenotype that would be present in the Nor-1 transgenic mouse model. This study demonstrates that the skeletal muscle reprogramming associated with improved exercise capacity and lean phenotype in the Nor-1 transgenic mouse model is associated with cellular recycling and autophagy. In summary, this demonstrates that Nor-1 expression in skeletal muscle drives the autophagy-dependent beneficial adaptations associated with exercise. And the study suggests that the nuclear receptor Nor-1 may be pharmacologically manipulated for the therapeutic treatment of lifestyle-induced chronic diseases associated with decreased exercise capacity and obesity.